Okay, good morning, everyone. Welcome to Grand Rounds today. Before introducing our speaker, I just wanted to make one announcement. The Chairman's Collaborative Pilot Awards are now accepting applications. They are due February 16th by 5 p.m., and they are open to all faculty of rank of assistant or associate professor. So our speaker today, Dr. Wendy Chung, is a clinical and molecular geneticist and the Kennedy Family Professor of Pediatrics and Medicine at Columbia University. Dr. Chung received her bachelor's degree and her medical degree from Cornell University, and then her PhD in genetics from Rockefeller University. Dr. Chung directs NIH-funded programs in human genetics of obesity, breast cancer, autism, as well as birth defects and pulmonary hypertension. She leads the precision medicine resource of the Irving Institute at Columbia University, has authored over 250 peer-reviewed papers. Dr. Chung is also renowned for her teaching and mentoring. She received Columbia's highest teaching award, the Presidential Award for Outstanding Teaching. In addition to our general interest in Dr. Chung's work, I also wanted to note that enhancing our expertise in human genetics and precision medicine was highlighted in the department's strategic plan. So we're all very interested in hearing what Dr. Chung has to teach us.
Thanks. So today, hopefully, we'll be able to tell you about some things that I've been doing, as well as some things that are hopefully a resource to the community here. Let's see. And with this, autism to me has been very interesting, almost from a sociological point of view, to watch how it's evolved over time. We've changed the definition a little bit, and I put this up here I know you all know the definition, but one of the things I want to point out in this is that as this has changed with the various DSM definitions of this, one of the things I think the public has sometimes failed to realize is that what this is and what this isn't. And I think there are a whole lot of things that get lumped into the autism umbrella. From a sociological point of view, I think they do it for very good reasons, which is that you get better resources if you carry a diagnosis of autism than if you carry a resource of neurodevelopmental disorder or intellectual disabilities. But I am going to come back to this a little bit because these are the core features that we do see in autism. And as we've been studying it, there's a lot of overlap. So when we think about autism, and I know many of you know this, it's an incredibly diverse spectrum, and that's one of the things I love about it. But it in fact becomes difficult to study from a research point of view, the thing of autism, because it's not just one thing. And so a lot of us over time have tried to study it. We've tried to get deep into understanding the brain by imaging, by functional imaging, look at it, how it evolves over time developmentally. And it really, to me, has been very difficult to replicate, to be able to have really rigorous and replicable findings, because in fact, it's so many different conditions. And if you take a slice, if you take, for instance, some of the very elegant neuroimaging studies that have been done, they're hard to do. And if you happen to randomly pick 25 individuals with autism, and then you 6 months later, pick another 25 individuals with autism, it's a very, very different group because it's so heterogeneous. And so that was one of the things we started trying to think about with this. One of the other points I want to make is that, and I'll get to this as I show you some of the data, although we categorically make the diagnosis of autism, we either say yay or nay, you do or don't have autism, I would make the strong argument that really it's more, as I think about it, a quantitative trait. That is, you can think about some individuals, if you think about from the social dimension, individuals, for instance, they may not make the official diagnosis by DSM criteria, but yet they've got some of the features. And in fact, when you talk with families, they'll often say, yeah, Uncle Bob, he's a little Asperger's, like he's got a little hint of the autism. And as we'll go and as we do measures like SRS and measures quantitatively in terms of social responsiveness, I would argue this idea of having a defined cutoff is really somewhat artificial. We use it for lots of good reasons, but still I would argue from a research point of view it's somewhat artificial. So The other dimensions that I want to highlight in terms of some of the diversity is, besides autism, many individuals with autism don't just have autism as a challenge in terms of social communication or social interaction or need for sameness, but they've got all these other associated features. And one of the challenges is that not everyone has the same other associated features. And depending on what your other associated features are, you may have other challenges. So if you've got ADHD, for instance, you've got one set of challenges. If you've got epilepsy, you've got a different set of challenges. If you've got gastrointestinal problems, you've got a different set of challenges. And so again, in terms of the burden that this places on individuals and families, it's not just the autism diagnosis, but it's everything else that's associated with that. So with all of that, and to be able to first start this by understanding from a research point of view, and eventually to change this into, or to translate this into what we can do to support individuals, many of us were concerned about this heterogeneity issue. And so the question became, can you get this more homogeneous? And I would argue homogeneity can come in multiple different dimensions. There are different challenges, I would argue, between males and females. There are different challenges across the age spectrum, different challenges we have in the little ones than we have in teenagers than we have in young adults. Different challenges in terms of severity, however you want to measure or call severity. And so again, trying to get homogeneity around that. And of course, as a geneticist, I'm going to be talking to you about some of the homogeneity we can talk about in terms of from a genetic point of view. In addition to that, and this is a question that parents or families ask me all the time, is when we think about etiology from a public health point of view, can we do anything to try and minimize the burden? And so part and parcel of the question also became, in terms of etiology, can we actually try and get more homogeneity by getting to the root cause, by talking about the etiologies with this? And I'll give you a sprinkling, a sort of high-level summary of what we think about, at least timestamp it today, January 31st, 2018, what we think about the etiologies. But I'll say, quite frankly, we really don't know in terms of most of the etiologies for this. I will talk about the things we do know, and that's not to say because they're the most important, but I do think that allows us to have some cornerstones, some really good scientific data to be able to say certain molecules are involved in the brain and the brain function. That's grounded on the genetics, but it's not to say from a public health point of view that those are all the answers or those are necessarily the levers in terms of how to minimize the burden going forward. We do worry a lot in terms of the intrauterine environment, the early brain development, and so we focused a lot of our ideas or a lot of our questions in terms of understanding exposures, whether they're things like viruses, infections, exposures in terms of medications, toxins, but from an epidemiological point of view, trying to get good study designs to actually address those very well is really challenging. To be able to have very large cohorts, again, without the bias that we have with retrospective recall, it's very hard to design those studies well. Some of you have been involved in some of the studies in the Netherlands that do this. I think the best that can be done, but they're still not trivial in terms of doing this. So let me start by thinking about one dimension where we see some of this heterogeneity, and that's around the dimension of gender. So autism is interesting. As many of you know, just to throw out a factoid, as my son would say, 1 in 68 individuals, based on the most current CDC numbers, are diagnosed with autism. So from a public health point of view, it's clearly very, very prevalent. And there's definitely a gender bias. So if we take individuals across the entire spectrum, we'll see about 4 to 1 boys to girls in terms of autism, or males to females. If we get in individuals at the quote-unquote higher functioning end, the diversity is more like even 8 to 1 in terms of that. And so one of the questions has become, what is the difference in terms of that? Why do we see more males than females? Could there be some sort of protective factor in females? If we think about, again, this as a quantitative trait, do females, are they shifted to the right in some way? Are they protected in some way? And if we have the same threshold that we use for boys and for girls, do we have a smaller percentage of females that meet those criteria? I don't know. We've thought about it from a genetic point of view. We've wondered, are there genes on the X chromosome that would account for those differences? And I'll give you the answer to that one. It's definitely not mostly the X chromosome, a little bit of enrichment, but that's not mostly the answer. I'm going to throw out something provocative and to say that maybe there are some differences both in terms of who comes to clinical attention. So in terms of boys, for instance, that are acting out, that are externalizing those behaviors, sometimes I think those are more obvious to teachers, to educators, to parents and they bring them to clinical attention. I also think there are some differences in terms of norms in society, in terms of what we are willing to accept in terms of that. And I'll also throw out provocatively, many of the researchers as well as clinicians in the area doing these evaluations are actually women. And there may be some differences in terms of gender norms and what women are perceiving in terms of this. But for all of those reasons, there could be differences within this. And just to say that I don't have the answer to this, but it is something that we continue to think about and to wonder about. So getting into specifically, because I'm going to spend a lot of time talking about genetics, what right do I have to talk to you about genetics? At a high level, what data can I show you to demonstrate to you that at least for some individuals there might be a genetic component to this? So I always start with this question of looking at twins. That always helps sort of right me in terms of whether or not a condition in fact is genetic. And I actually think this is a very interesting number. So if you take identical twins, so genetic, monogenetic twins that are identical as far as we know in the same intrauterine environment with the same genome pretty much, very high concordance. So if one twin has autism, about 77% of the time the other twin will have autism. But what's remarkable to me about that number is it's not 100%, right? So other conditions where it's absolutely cystic fibrosis, sickle cell anemia, things like that, that number is spot on 100%. If one twin has it, the other twin has it. And so this suggests to me that it's not all genetic. It's not all nature, if you will. There's some nurture that's involved with this. But yet, when you also look at other dimensions of this, and so this is, again, against this backdrop of the general population being about 1%, there are some other factors that are going in that also suggest to me that it is a combination of both genetic and environmental factors. So again, another factoid, because this is something that parents come to me with a lot of the time. If you have one child with autism, and as parents you want to have another child, there's about a 20% chance that your next child will have autism. That number, I can tell you, for many folks is a scary number, and in fact, that's why they come to me in this business, trying to figure out whether or not I can tell them if their next child will or will not have autism. But another interesting factoid is compare this between fraternal twins that are sharing the same intrauterine environment to the siblings. So siblings in this particular case genetically will be 50-50 in terms of sharing 50% of the genes. But again, what's different about fraternal twins is they're sharing the same intrauterine environment. What did I just do? Same intrauterine environments, and also to a large part the same sort of early life stage exposures in terms of this. And this again suggests to me that it's not all about the genes. There are some shared environmental features too. And I can't absolutely prove the burden in terms of how much is prenatal versus postnatal, but I do think this is telling us that there are some non-genetic factors as well. But again, it's a combination and probably different people's sort of different contributions. So one factor within this that I think took us as a community a little while to realize is the following. So many people will say, well, it's genetic, and so anything genetic must be passed down in the genes from the parents. That's the way genes work, right? And in fact, that's not entirely the way genes work. And when you think about this, if you think about individuals, think about them at the quote-unquote severe end of the spectrum. So individuals in particular who are nonverbal, who might not grow up, for instance, to get married, to have children. If it's genes, if it's in the genes, how in the world do those get passed down? From a population point of view, how do those genes exist in the population if they can't get transmitted generation to generation? And so that brought up the idea maybe some of these genetic factors were actually starting brand new in the individual with autism. They, in fact, weren't being passed down from either the mother or the father. These were de novo genetic factors. And so with some new technology, the new technology being the decreased cost of sequencing or other genomic analyses, it suddenly became possible to do an experiment we'd been wanting to do for a long time. We just didn't have the NIH dollars to do it. And that is that if you actually now go back and genetically compare the mother and the father with a child with autism, and when I mean compare, I mean you sequence 3 billion letters of their genome and figure this out, you can in fact determine what's different between that child and both of their parents. So again, I can say that I've done this experiment myself, and this is just simply to show you that this is over time on the x-axis, and on the y-axis, the logarithmic cost of sequencing. That sequencing cost has come down. So first genome that we sequenced cost us $3 billion. The last genome that I sequenced cost me about $800. Tremendous decrease in terms of price. Still cost me about $5,000 to analyze that genome, so it's not quite trivial yet to be able to do, but you can start to be able to get your arm around these things. This was still expensive, so many of us have taken a shortcut in terms of analyzing the genome. We don't analyze all 3 billion base pairs. We'll look at the fact that this is what your genome looks like, and for those of you who had enough coffee this morning, you may not see this, so I'll help you. Within our genome are little gold nuggets of coding regions, the parts that actually get translated into the proteins and other coding regions of the genome. These comprise about 1.5% of your genome. And so for some of us who are cheap, which are many of us on NIH budgets, instead of sequencing the entire genome, we'll take the shortcut of analyzing just these exons or the exome as we're doing it. And so we started out by taking that as a shortcut on literally at this point, it's been tens of thousands of individuals, and we're up to about 10,000 individuals with autism in whom we've done that. And we've analyzed these between, for instance, the mother, the father, and the child, and looked for these new genetic changes in the coding region. Now, if you actually did this on your kids, and I can admit that I've done this experiment on my family, if I analyze and look at my kids and look at their genetic material between my husband and I and my kids, there is on average one coding variant in your genome that is new from your parents. And so by doing this, we have now simplified this from an analysis point of view. Instead of having to look at 20,000 genes, instead of having to look at 3 billion base pairs, I'm essentially looking at, on average, one variant per individual. And that now becomes something that you can actually get your head around in terms of understanding that. So that was a basis in terms of being able to analyze and come up with a lot of the data that I'm going to be talking to you about. But let me try and reassure you that is just one piece of the puzzle. It's a very important piece of the puzzle, but it is just one piece. And in particular, the piece of the puzzle, this is the dimension here that I'm trying to show you. This is the frequency of a particular variant in the population. These are very rare variants. These are very common variants. And then this is how high the impact is. And so these are the impact of variants up here in this quadrant is very, very large. As I say to my medical students, things up here, these are genetic, really, really powerful genes, where it's really mostly the genes that are determining things. And then these variants over here that are very common in the population but have very low impact, those are genetic. I mean, those are things that really any one variant is not able to predict in terms of what your behavioral phenotype is going to be. But on the other hand, what we're starting to learn is you package these together, you look at 100 of these genetic variants, that can now start to predict what we're talking about in terms of this. So with autism, the thing that's challenging is we call it all autism, but in fact, there are some individuals who have these very powerful genes, and there are a lot of people, though, that have these, and there are probably people in the middle here that have a combination of powerful, sort of intermediate in terms of frequency, but we call it all the same thing. So that's what we've got to quit doing. So we've started doing this in terms of looking now at many of these children through the technologies that I showed you about, and this is just to show you that this is actually exploded. This is just over a couple of years, all the publications we had in different genes that we've now defined as autism genes, and to say we're guesstimating that the total number of these is going to be about 500 of these very powerful genes. And just to give you a scorecard, we're probably about 100 into that. We probably know about 20% of those genes so far. So let me give you a couple examples about how that can be useful. So this is actually, to me, one of the purest genes in terms of really being associated with the core features of autism. It's a gene called CHD8, and you don't have to worry too much about it. But the interesting things about it is now instead of this individual with CHD8 having all of these other associated features, you can now start to see very, very specific, if you will, subtypes of this. So individuals with CHD8 very, very characteristically have very disturbed sleep in this. I mean, I have kiddos with this that, for instance, will go days without sleeping, much to the chagrin of their parents in terms of doing this, but sleep is a very, very fundamental part of how the brain is different in this case. And the gastrointestinal problems, you can literally do physiologic experiments to see how much the GI transit is slowed down and how much that actually disturbs them. But these individuals are actually different in the sense that in terms of intellectual disability, most of my kiddos with this will have IQs 90, 100. These aren't the kiddos with IQs of, for instance, 40 or 50. On the other hand, there are other genes, an SCN2A, a sodium channel in this particular case, very, very interesting to me, because for those of you who are interested in this, we can actually have for this sodium channel both what we call gain of function mutations as well as loss of function mutations, different amounts of sodium that are going through these pores that change the currents, and we actually can have, depending on whether it's a gain or a loss of function, either autism or epilepsy, but they seem to actually be reciprocal in terms of that. It's not absolutely perfectly the case. There's some overlap, but we tend to see that. And in fact, one of the interesting things is in terms of potentially therapeutic insight, these are some of our more druggable targets. Being able to do this, there are lots of ways that we've developed in terms of things to be able to modulate channels, either in the brain or the heart. And so potentially this is one of the areas that from a therapeutic point of view, there may be great insight. On the other hand, for these individuals, They have much more in terms of, for instance, lower IQ, much more problems in terms of intellectual disabilities. And so again, depending on this, it helps us in terms of prognosis, and I would argue at the end of the day, it's going to help us therapeutically as well. So I wanted to bring to you news you can use. So what's actually, not just what's pie in the sky in terms of esoteric research we're doing, but what can you actually use in clinical care? And so this is whether you happen to be interested in autism or you could be interested in any other neuropsychiatric condition. This is from the trenches. So this was actually clinically, in clinical care, when we use this technology of exome sequencing, by the way, covered by insurance. So this is not, again, some sort of fancy genetic research study. This is in the trenches of what we're doing. This is the yield on the y-axis here by a variety of indications. And I don't expect you to be concerned about all the indications, but I've circled autism here. This was often, just to give you a sense, a 5th, 6th, 7th line genetic test. So this was after many people exhausted many other tests before, and still the yield here was 14%. If you do this as a first-line test, I can tell you the yield runs about 25%. What does it tell you? It tells you about 25% of the time you'll find an answer and 75% of the time you won't. Why don't you find an answer 100% of the time? Well, in part, it's not all genetic, as I told you. And in part, and importantly, and I'm modest about this, we don't understand all the genes. Right now, we understand about 4,000 of the 20,000 disease genes, but every time I go back and actually look at this data again, year after year, I'll find about another 3 to 5% of cases that I can now interpret and now understand because our knowledge and our understanding of the genome is improving. So, and this is true, and I won't go through all of psychiatric conditions, but this type of yield is reasonable. There's some yield that comes with schizophrenia, for instance, with the same type of test. It tends to be the case that the more this is associated either with the more severe end of the scale, the younger the age of onset, the more it's associated with other medical problems, the higher yield with this. And again, this is something that is very clinically readily available if it's helpful. So as we've done this, it's been an incredibly interesting journey for me to take from a sociological point of view. This was actually one of the first genes that we described associated with this. For those of you that are interested in the molecular biology, what's incredibly interesting to me is that there's in this one gene, there is exactly one and only one mutation that we see. Even though it's the recurrent mutation that is new in those children, we see only one place in this gene that you can disturb and cause this problem. It's this one particular missense mutation. And for anyone who's interested, I'm glad to talk to you about later, but it's because it's probably associated with a gain of function mechanism in terms of this. What's been interesting about this is a couple things. Number one is my universes have collided. Although I do a lot of work in autism, I also do a lot of work with congenital heart disease, and as I'll show you in a second, there is actually significant overlap in the biology of just how you form a body, whether it's how you form a brain, how you form a heart, many of the same mechanisms involved in that, though. And this is with permission of the families to do this. One of the interesting things in this is just the self-organization of the families and the communities themselves. And so even though these are very, very rare conditions, this was a family meeting we had last spring. It was the first time the families got together, and only in the Facebook era could this happen. But within this, we now have almost 100 families from around the world who have aggregated around Facebook to find each other, to learn from each other, to exchange information between doctors and patients. And this has really led to a completely different era in terms of patient engagement for research as well as for their clinical care. So this is just to show you sort of a Venn diagram, as I was saying, in terms of, for instance, how some of these genes are overlapping. It is amazing to me how many of the genes, for instance, from intellectual disabilities, congenital heart disease, epilepsy, schizophrenia, it is now becoming increasingly apparent how much overlap there is between that. Even within specific genes, different variants can lead to one or another phenotype in terms of doing this. And this increasingly has, I can say, incredible significance in a very interesting way, because we are starting to detect these things even prenatally. So as we're doing ultrasounds, for instance, on pregnant women, we are oftentimes seeing the structural birth defects and now starting to do tests like exome sequencing in the prenatal setting, starting to make these diagnoses, and in a very, very interesting way, giving potential parents a huge amount of information about prognosis and about other behavioral or psychiatric challenges that may be in the future, and incredibly interesting to understand how they're dealing with that information, making decisions, and ultimately what the impact is in terms of parents and as they bring up their children. Just a little bit about the biology. There are several different types of genes that we're consistently seeing: histone modifiers, transcription factors, even DNA binding proteins. We're now getting into the non-coding sequence and understanding both long non-coding RNAs, microRNAs, but we're seeing many, many sort of systems in terms of systems biology that are converging on these. And for anyone that's interested in this, we now have so many different projects that can come out of this basic discovery efforts that we literally can't keep up with all of this. As we're doing this, it is interesting that we're using a lot of developmental biology to understand this. So we can look at, for instance, genes that are expressed in a developing heart. Those genes over to the right side are expressed at very high levels. We can look at chromatin modifiers as a class, and most of the genes that we see in congenital heart disease we now realize are actually up in this quadrant here. But also, if you look at this, many of these are also, as I said, the same genes. If I were to make this an axis about the developing embryonic brain, it's those same genes that we're seeing in terms of the brain as it's developing. So let me just give you the scorecard in terms of, at least right now, where I think we probably stand with this. Interestingly enough, it's probably about 10 to 30% of individuals that have de novo genetic events. The individuals that fall into this bucket tend to be the ones that are, on average, on the more severe ends. They have more problems in terms of intellectual disability, lower IQ. They may have problems in terms of other medical issues specifically. I don't think we haven't had the sample size yet to be able to catalog all of these, but our estimates based on heritability is probably about 50% of the answer, though, is due to inherited genetic factors, probably not in any one family one factor, but as many as literally tens or even up to 100 of those. The environment also playing a very important role in this, and I'll just say very briefly, this idea started out with Chris Walsh, actually, but even the idea that there might be somatic mutations that are involved. And so the idea here is that even after conception, after the egg and the sperm unite, and let me go back to one point I should have made, that these de novo mutations, some people ask me, where do they come from? Why do they exist? Is there anything we can correlate with that? And I have to, just so that you can educate if your patients ever ask you, there is an association with advancing paternal age that goes with that. And so one of the associations that people have made epidemiologically in terms of older fathers with autism, at least a significant portion of it is due to the higher frequency of these de novo changes as dads get older. You can get de novo mutations that way, but another way you can get de novo mutations is after the embryo has actually formed, after the egg and the sperm have united, there can be genetic changes that happen after that point of fertilization. And in those cases, it's interesting because the developing embryo and eventually the human really are a combination of different cell types, some that might have the mutation and others that might not have the mutation. And depending on what cells in the developing body have it, you will have either greater or lesser manifestations of that. And it ends up being difficult to make these diagnoses because if you can imagine it, if we're doing a blood test, for instance, to make the diagnosis, and these mutations are only in the brain, where you're having the manifestations, I'm not going to see it in the blood. The only place I'm going to see it is in the brain. And so the reason why Chris Walsh was actually very successful at doing this is literally at the time, for instance, kids were having surgery for epilepsy, medically intractable epilepsy. Some of the surgeons were literally taking out pieces of the brain, and you could directly look at that. And we've developed some better ways of doing this in large part on the backs of cancer biologists who had done this looking at tumors, and tumors have a lot of heterogeneity of cell types and mutations. And developing some algorithms with that, we've been able to have greater and greater sensitivity to detect some of these de novo mutations, either in blood or in other tissues. At this point, and again, very high-level summary, our best guesses are probably somewhere about 3 to 5% of autism are due to mutations like this. And if anyone's interested, and I can tell you exactly how you make these diagnoses, but there is some of these really hard to diagnose cases that are due to these types of changes. Okay, so let me take a deep dive for just a few minutes and show you an example of where we decided to take just one genetic form of autism and then go as deeply as we could think about doing it and trying to understand how the brain worked, brain function, brain structure, how these things tied together. And so when there was a cohort that was created by the Simons Foundation, something called the Simons Simplex Collection. We were a site here at Columbia for that. Out of those 2,600 or so individuals in the SSC, the most common genetic form of autism was due to a copy number variance in this chromosome region here, chromosome 16p11.2. About 1% of the individuals in the cohort had either a missing or extra piece of that. And I've just shown that region here. There are 29 genes that map to the region, and we decided that would be something to try and understand the brain better with that. So we did this in terms of a deep dive. We found people around the country, around the United States, that had this particular copy number variant, and we invited them to come to some of our clinical sites around the country, and we basically tortured them for 2 to 3 days, depending on how quickly they could go through things. We put them through all sorts of psychometric measures. We interviewed their parents. We put their heads into to MRIs. We did MEGs. We did as much as we could, and then we also collected some biospecimens. And one of my public service announcements here, unabashedly as an advertisement, all of these data, all of these biospecimens, free to anyone here who wants to use them. So if you can think of a better way to use them, please tell me, and I'll be glad to give you everything. As we did this, again, this was powered because more patients were finding out about their diagnosis through clinical care, and we figured out ways of rounding them up through the internet, through Facebook, and as we did this, created a community for this. We've now got over 600 families in the community. As we did this, we had these great opportunities to meet with families and educate them about what we were doing as we went along this journey. And as we did this, the first studies were based on 100 individuals, 100 individuals with the deletion, 100 with the duplication. And to us, that was still a relatively modest number, but at least we had the homogeneity going for us as we did it. Let me just go quickly. We had these people scattered around the country as we did these studies. And again, these are about the 100 that we have, split approximately 50-50 males to females. And across the age spectrum, skewed towards the younger ones. We were especially interested in understanding early childhood development with disorders like this. So some high-level things. What can you take away from the cohort in terms of this? Well, one thing that we saw very quickly, and deletions here and duplications here, so having a little less or a little more of this particular region. One of the most interesting things we saw right away is what we call the mirror phenotype. That is that when we saw individuals with deletions, they tended to have larger heads, individuals with duplication, smaller heads. Not absolutely, so in terms of actually, being 2 standard deviations above or below the mean. Individuals didn't necessarily meet criteria for micro or macrocephaly, but definitely shifted in important ways. I'll come back to it again, but we see specifically obesity in these individuals, I think centrally driven in terms of ingestive behavior and appetite. And as we got deeper into this, for instance, we actually looked then at their brains by imaging. We did volumetrics, actually looked at controls. In this particular case, controls were familial controls, either parents or siblings that weren't unaffected. With the deletions, very obviously, it wasn't just that they had, thick skulls. They literally had bigger brain volumes or smaller brain volumes for duplications that we could see this. And if we went through systematically and went through either the white matter, the gray matter, region by region across the brain, essentially is a universal thing. We're not seeing any one specific portion of the brain. It looks like it's a global effect in terms of how the brain's developed. That very consistent in terms of that. So we tried to start looking at function. And so as we did this, we started thinking about, can we look at a very sort of primary things, looking at sensory input and how that's recognized or how that's received. So we did these where we played a ringtone for the kids. We'd be able to say ba, literally a ringtone, ba, 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 and have that change, and then be able to, with Meg, see exactly how long it took for that to register in the brain. Was there a delay in terms of any of those synaptic connections or the primary sensory detection? And so in fact, and this is looking at usually in a normal individual, you would receive that signal at about 100 milliseconds. In fact, with individuals, especially with the deletion, what you can see is you can see that shifted. It actually takes longer for them. And if you plot this out in terms of this bar graph, you can see very specifically, even just for primary sensory input, we already see a delay in terms of the transmission of this information. Very, very high fidelity in terms of seeing direct outputs from that. We started looking then at just some of the things that the families were reporting to us, and then we did it in a more sort of structured way. So we started looking at whether people were right-handed or left-handed to try and understand in terms of specialization of areas of the brain. And one of the things we quickly saw is that here are the controls here in blue. Our individuals in the red here with the deletion actually much more likely to be ambidextrous or less specialized, less concentration of specific functions in specific areas. So again, even in a very practical way, we can start to see some of those differences. And as I said, problems in terms of micro or macrocephaly, but also problems that we were seeing in terms of seizures. So a significant fraction, about 25% of them had epilepsy as well. And I won't go into this in too much detail, but lots of, in terms of neurological exam, differences in tone, differences in terms of gait, motor coordination, motor apraxia, problems with speech in particular as a delay in that. So we saw lots of consistent features. But one of the things that actually got me at the very beginning of this journey was, remember that I told you that we picked this particular copy number variant because it was the most common genetic form of autism. Now, in this particular case, we did what I call a genetics first strategy. So we first picked people because they had the 16p, either duplication or deletion, and then went back to them and saw what their behavioral manifestation was. And in particular, when we did this, we ended up finding that about a quarter of them had autism, but in fact, not everyone had autism. And even individuals that were old enough that they should have had autism by that age if they were going to have it, did not have autism. But they all had something, I would argue. So different of them, for instance, and many of them had more than one problem. So some of them had autism, others had phonological processing disorders, others had developmental coordination disorders, others had problems in terms of expressive language. So all of them had brain differences, but they didn't all necessarily meet ADOS or ADI criteria for fully having autism or having problems in terms of social communication. So we started thinking about this more as this quantitative trait that I was alluding to before. So within this, then, it was important to me because people started saying things like, well, this is a disorder that's incompletely penetrant. Not everyone who has the gene has a manifestation. This is something that's not sort of one-to-one connected. And I would argue that's not true. It's a matter of how you want to define what the impact is. So if you now convert this to, if you were to say, for instance, it's incompletely penetrant for autism, I'd say you're absolutely right. Only 23% of individuals have autism. On the other hand, if you want to take this, for instance, with a quantitative trait like IQ, and if you do the IQ very rigorously like we did, and especially if you compare them to their familial controls, so if you compare them, for instance, to their siblings or to their parents that don't have the deletion in this particular case, this is what it looks like if you have 16p. In these individuals, and this is what it looks like for their siblings and their parents, and on average, their IQ is shifted almost 2 standard deviations lower in terms of IQ. Now, if we were to define this as intellectual disability with an IQ less than 70, most of them are not intellectually disabled, so they would be incompletely penetrant with that definition. But again, I would argue that they all have taken some sort of decrement in terms of their IQ from their predicted familial IQ. And it's not just in terms of that. If we now convert autism into a quantitative scale of some sort, in this case using the measure of social responsiveness scale, in this particular case, higher number means more autistic. Again, here are the parents, here are the siblings, here are the individuals with autism, or individuals rather with 16p. And again, in this particular case, shifted, it's actually upward with the way this scale works, but about 2 standard deviations closer to problems in terms of social responsiveness. And so we've done this with many, many different things. We've done this with fine motor skills, again, see about 1.5 standard deviations in terms of a decrement for this. Even looked at body mass index, but all the measures that we've looked at, essentially we see the same thing, that there is quantitatively a shift or a difference in this to the detriment of these individuals, but it's not necessarily that they're meeting an official criteria for how this comes about. So one of the first things we're actually going to be doing with them is dealing with one very practical issue. So one of the things that happens with these individuals is that when they become teenagers, they start actually gaining a lot of weight. And for many of them, with many of the behavioral problems they have, they get treated with medications that actually exacerbate that. They have more problems then with metabolic control, have much more prone to diabetes, even with the same amount of adiposity. And this is at a time, again, when they're in adolescence a lot, when they're gaining the weight. So one of the first things we're going to try doing, potentially, is being able to deal with that very pragmatic issue. And this actually comes from the neuroscience of understanding with some work that we did a couple decades ago, starting to understand the circuits in terms of in the hypothalamus, where we have centers for ingested behavior and understanding satiety. And I won't go into this in a huge amount of detail, but to say that we know that there are signals coming from the peripherally. They're actually having their signal or receiving their message here within the hypothalamus on first and second order neurons. And eventually the signal is coming through the melanocortin receptor. That's actually what's ultimately helping to set the set point in terms of what your body weight is and being able to feel full and being able to do the balance of food intake and energy expenditure. And so with this, there's a company that has developed actually a medication, which is a melanocortin 4 receptor agonist, which has the effect of being able to make you feel full. And as they've done this, they've been thinking about the specific molecular defects that they know flow through this system, through the melanocortin 4 receptor, and that includes some very, very rare genetic forms of obesity. I will emphasize rare at this point. But here's one of the first, this was actually published in the New England Journal of Medicine last year. Here are, to me, really, really stunning effects. So these are individuals that have a rare form of obesity due to a pro-opiomelanocortin receptor mutation on the Red here is the hunger scale, and here is the weight. You can see almost instantaneously after starting this, they feel full. They no longer feel hungry, and you can actually see the weight loss. This was a clinical trial that went for 14 weeks. As soon as that ended, and as soon as they stopped the medication, immediately her weight, her satiety, or her hunger rather, go right back up. Thankfully, they started her on an open label extension, came down again, and just a remarkable impact in terms of that. How generalizable is this? I don't know yet, but one of the things we've started thinking about is for other individuals where at least the molecular biology suggests that it's going specifically through this pathway, will we be able to alter this in terms of being able to ameliorate some of the problems that we've had? So I don't know the answer. This is actually, we're just starting this clinical trial, and we shall see. But the last thing I want to end on is one last public service announcement / unabashed advertisement, and that is that as we've realized this, we've realized it's a numbers game. That to power the research for the autism community, we just needed to be able to get more people engaged in research, and we needed to, as much as we could, to be able to define specific subtypes of autism to go forward. And so this is an initiative I started actually April 2016. We call it SPARK, Simons Foundation Powering Autism Research for Knowledge, and it is funded entirely by the Simons Foundation. And the goal of this is to really create a community of at least 50,000 families that will be research available, either in person or online, to be able to do this with the dimension of genomic characterization and with real patient engagement, with real engagement from the community. And again, it is freely available to anyone that wants to use this as a recruitment method, to recruit for their research studies, to use the data, to use the genomic data, to use as a comparison cohort to any other psychiatric condition you might be studying, but it's meant to be able to be, as I said, a resource for the community. We actually have, it'll be coming out in a week, and NeuroViews will be in Neuron, will be in a very detailed description of this entire cohort and what it involves. So please go there in terms of being able to see more about this. But it has been tremendous to see just what the response rate has been from the community in terms of doing this. As we've done this, let me just see if I can quickly get through this. As we've done this completely online as well as having clinical sites. The Cornell has a clinical site in Westchester at CADB to do this, but it is meant to be a registration that can be done online. The whole registration takes about 20 minutes to be able to do. If people want to do the DNA part of this, the genetic part of it, they can give a saliva sample from the convenience of their own home. And one of the things that I put in within this is to be able to get those families this information back. So if they, for instance, fill out a questionnaire or one of the instruments online, they actually get their data back. They can see how they scored or how their child scored. If they do participate in the genetic part of this, which includes exome sequencing as well as genotyping, they actually get clinically relevant results back to them at absolutely no cost to them. So it enables them to really be partners within this whole process. And just as an example, these are where we are around the country. And just to be able to say, in terms of what we've done, within the first 20 months of doing this, we've already had 30,000 families be able to sign up and fill out all the registration questionnaires, consent, be able to supply their DNA samples, and we've started providing results to individuals. And as we started actually doing what we call the research match, that is having researchers use this tool to do this, we've had uptake rates of over 60% for online surveys that patients have actually engaged and done this. We've been closing out recruitment for many different clinical trials around the country and studies within a month. And so it really, I think, has what has been very difficult for people to be able to get their studies done. We think is going to be a catalyst to be able to get this done. And again, as a nationwide network, even for consortia or for research communities that have multiple sites in doing this, a way of being able to power those studies. So as we do this, we have done this very much with patient engagement. I love what we've done. This is very much, I mean, it's so 2018. We're on Twitter, we're on Facebook. As we do this, we have webinars, we have Facebook Live. We put out questions to the community so they help us answer the questions about what's important. We put out questions, what are important, what do you want researchers to know? As an adult with autism, what do you want others to know about? What are important things to you? What are questions that you've been wondering about? Once a week, we put out questions like that, and as we hear back from them, we actually get a much better sense of where we should be driving the science and the gaps that we should be filling with the community. So all of that's been great to be able to see, and in the interest of time, I won't go through all of this, but anyone who wants to is welcome to it. The most important part of it, let me just get to, is hopefully the URL. here, which is anyone who would like to have any of the information, be able to use any of this information, any of the data, any access to any of the participants, just go to this URL here. And if you have any trouble navigating this, just send me an email because we want this to be used by the community. This is going to be a longitudinal registry. Again, we want to get to at least 50,000 families. The original goal was within 3 years to do that, but we've got a 20-year commitment to maintain this registry. So this will not be just a short-term thing. This is meant to be long-lasting in terms of being able to use this for the community in the long haul. So as just in summary in terms of doing this, I know this is a complicated condition, but we are trying to chip away at it piece by piece. It won't come overnight in terms of doing this, but we do hope that genetics will be one portion of being able to disarticulate it. It's not going to be the only dimension, but I do think it allows us to give us some real strong anchors in terms of understanding the neurobiology. And as we do this, I hope this will be an example of how we do more things that make them patient-centered and really engage the participants in a very meaningful way. So I will stop there, but just to say that all of these take tremendous teams, and I'm very privileged to be able to have great colleagues to do this with. These are members of our Spark team here, members of our Simons VIP team here, scattered throughout the country, and I'll be glad to take questions either now or by email later. Jeremy?
Sorry, for me, I could just holler, but we try to use the mic, apparently. So there are lots of things that we could ask you more about. That was really a wonderful talk. I wondered, you made a couple of sort of what you described as provocative comments on the question of sex early. And I wonder if you could comment, a lot of the rare variants, de novo variants, that have been described in big enough populations to be able to comment have fairly even sex ratios.
Yeah.
And that, coupled with the observation that you see more even sex ratios when you look at baby siblings who are deeply phenotyped quite early, not necessarily just when they would ordinarily come to clinical attention, but for screening and largely in the research context, those sex ratios are also a little closer, although it's more like 3 to 1 instead of 4+1. I wonder if you can comment on what you think that might mean and how we need to think about sex differently when we're thinking about which kids we need to ascertain or screen in the clinic in the first place?
Yeah, so it's a great question. I think there are actually two different answers to both of those things. So, and I apologize, but for the aficionados in this, so many of the genes that have the big impact for those, they're not on the X chromosome, they're on the autosomes, and so we don't see any gender differences between that. But they're really, so if you look at the sort of severe low IQ or nonverbal end of the spectrum, those tend to be enriched for those genes with very, very high impact, and they're gender neutral. They don't care about gender for that, and so that's why I think we see those equal genders within that portion of it. The baby thing, I think, is the ascertainment issue, and so I think that's the issue that comes that if you don't wait for someone to come to clinical attention, for whatever reason they get to clinical attention, if you really look very carefully, I think the males and the females actually have their own differences. We've just been using, I would argue, some norms that are more based on one gender and applying them to another gender, rather than having gender-specific norms to a certain extent, in terms of really digging deeply to understand the social connectivity, the need for sameness, all of those same dimensions. I would argue, and I don't know, those of you, I have 2 boys, so that may have sort of skewed me, but boys are just louder. They're more in your face, whether, they come to attention. You can't ignore them in some cases. Whereas a lot of the girls, I think, have a lot more internalizing behaviors, and I just don't think we're as aware of them, and we don't necessarily probe for them. If they're not screaming for help, we're not sort of looking for trouble all the time.
What a terrific talk, Wendy. Thank you so much. Just a small question, given that you did give environment its due. Granted, it's relative, but with this aim to get 50,000, you do have some opportunity to ask maybe some low-hanging fruit questions about the environment, maternal illness during pregnancy. Will that be part of the data collection? Yeah,
so great question. So in fact, our very first study out at the gate was something called GEARS. It was done by the group at Hopkins, and it was specifically focused on the factors during during pregnancy. So they're in the process right now of analyzing the data, so I don't know what the results are yet. But that was actually something that we heard from the community both of researchers as well as the community of participants, that they were very interested in that question and wanted to be able to dig deeply. And if anyone has suggestions, let me know, because from a funding point of view, I still think beyond anything we can do in Spark, I still think it's a very important question. It's just very difficult to design the study to get at the data. And I do know, and some of you have them, birth cohorts in a variety of ways I think can be done for this, and if people have good ideas about how to do it, there are funding opportunities to be able to do well-planned studies to address these questions.
Again, Wendy, that was just terrific. And to follow up on Catherine's point, I hadn't thought about this till right now, but the ECHO cohort, the new nationwide, that would be a wonderful group to partner with, because there is so much phenotypic information that's going to be gathered upon 50,000 individuals. And that to be able to pair that with some of the same things you're trying to do, I think would be a great resource. Right. And among other things, I'm wondering about, you had mentioned some of the factors involved in some of the de novo mutations, particularly regarding sperm and father's A. There we go. Are they picking up other information about other paternal exposures? For example, I've heard stories about alcohol exposure and how that can affect the incidence of these mutations, sometimes stress, pollutants. Is that kind of information also going to be gathered?
So that people are asking that question. They've actually asked it even across different studies to increase the sample size for different conditions that are associated with de novo mutations. None of them are coming up with a strong enough signal right now beyond paternal age. Paternal age is wiping out in terms of the contribution. Could there be a modest effect of some of those other things that we won't know until we get to sample sizes in the hundreds of thousands to millions? Possibly. But overall right now it's very, very clear the signal is coming mostly from paternal age, tiny little bit from maternal age.
Well, it was a wonderful presentation for somebody that doesn't really familiar with this field at all. So I want to ask you something completely out of left field, which is, does the fact of the complexity that you're beginning to unravel a little bit here and understand in more depth mean that the ethical concerns about people engineering their kids are so far off in the future we don't have to worry about them so much?
Okay, so that's interesting. So this is something that I actually get asked, I just got asked yesterday as an example. So the simple version of the engineering that you're talking about that practically people are doing. So many families, especially in New York, I have to say, with a child with autism worry about this risk of recurrence, and many of them will go through and they'll come to me saying, if you can find the gene, I want to sort of weed that gene out of my gene pool in some way. Practically, what many families are doing right now is doing gender selection. So they'll go through in vitro fertilization. They know the risk of recurrence is greater for a male than it is with a female, and they'll say, the only thing you can do for me right now, unless I find one of these things, and I'm very, very good at it, they'll say, okay, well, we'll do this, and we'll specifically have select for a female as opposed to a male, and we'll keep our fingers crossed. That's sort of how it's playing out right now. There are cases, I just had a meeting with a group yesterday in which we do find things where they are autosomal recessive. That is that there's a 25% chance of happening again. Those families will absolutely, very commonly, if they can afford to do it or their insurance will cover it, select against those. I don't consider that an ethical problem right now because those are really, really severely debilitating. We don't have any real good treatments for those. I think the question that people ask us that we say, no, we don't know the answer to this is, when it comes to many of those genetic factors where it might be a combination of 3, 4, 5, a dozen sort of things, can you select for or against those? As you said, I'm not going to worry about that problem in the next 5 years. That's realistically not where we have to be. But on the other hand, I will say, and this has actually come up already in Spark when we returned these results, I do end up in circumstances where we return results and we do have a parent that's oftentimes for an X-linked disorder, someone that carries it, the mother in that particular case that carries it, where the mother will have a milder phenotype. And I have to say there are some interesting just psychosocial things that go on then when we see transmission from a parent to a child. There's the blame game that goes on. There's the introspection in terms of, am I who I think I am? Is there something wrong with me? And one of the things we've been hearing from the adults in Spark is some of this concern about this genetic portion. And in fact, many of our adults that are, I call them self-advocates, they don't want anything to do with the genetics. They specifically will not participate because they don't want this to become a eugenics movement against individuals with autism. So it's a interesting and complicated question.
So I know in addition to all of this stuff on the molecular side, you do a lot of thinking, as this reflects, on sort of the ethical implications of this and the experience of the families. And I just was this weekend struck by several child psychiatrists saying to me, we had actually told them not to do pharmacogenomic testing in child psychiatry because it's completely useless. Anything that it'll show you will disagree with an FDA indication. In any case, they then equated that to, oh, we shouldn't be doing genetic testing, and they already have, unfortunately, in child psychiatry, a bias against doing this because they can't explain to families why they should be doing it.
Right.
And I have this conversation with families a lot, and some will, some won't. But I wonder if you could talk a little bit for this group about why we do it. We just had a piece on this with some of the folks at Yale about what child psychiatrists need to know to make sure that this happens. But I think it isn't just true in child, it's true for folks with schizophrenia and so forth. But what is the gain?
Sure, sure. And for anyone who's interested in it, I actually have webinars or videotapes online about doing this. I actually gear them to families as well as more lay audience, so they're very accessible cognitively. So there are a bunch of reasons for doing this. And I didn't include this slide in the deck, perhaps I should have. So one is that although I think it's the end game, we are not there yet in terms of the end game, clearly it is about tailoring care. It is about being able to improve prognosis. It is about being able to anticipate who's going to have problems with seizures, who's going to have problems that are medically treatable. I call it news you can use, so things that really you can impact. I would say there are specific use cases for this. I do think that as we're seeing, and I can, we share some of the same patients, some of my kiddos that are now transitioning into adulthood, they are very interested in understanding also some of the medical things they're going to have to worry about because they are not so good at accessing healthcare, and their parents especially want to set them up in terms of making sure they're prioritizing what they need to do in terms of health maintenance. It's not just in terms of mental health maintenance as well. So that's one of them. And I would say I can check off for you the cases in which it's helpful and cases in which, to be honest, right now it's not because we're still learning. But we've got to start that learning process, I would argue, to be able to get to the point where we've got that for everyone. So that's number one. Number two is for the risk of recurrence. And increasingly this is coming up not just for parents, but it's coming up for siblings and other family members. They've seen Johnny or Susie in terms of having autism. They say, I don't know that I should be having kids. This actually happened to me at dinner last night with a family. And being able to understand for siblings or extended family members at risk of recurrence has proven to be very helpful. In some cases, the risk of recurrence is 50% or 25%, but knowing that we've got reproductive options that do not involve abortion, very, very helpful for families in terms of doing that. Prognosis is very helpful. I know that, for instance, the other day I was giving a result for FOXP1 and being able to say, for instance, that language is a very particular problem for this and that we've got multiple ways of augmenting communication, other devices that we know that are very, very helpful, even though they can't get the words out right then, and being able to say there's a light at the end of the tunnel. They may not talk until they're 8, but they are going to talk, I think is incredibly helpful for parents in certain situations. I will also say on the flip side, being able to say in some cases that the prognosis isn't good is not something all parents want to hear, or they may not want to hear right now. So I think there's a balance that comes with that. The final thing I'll say, and this is really a cosmic thing, and different people have different cosmic looks at, views of this, is just there's this question of why. For some families, they really, really want to know the answer why, and I'll say disproportionately, I see more moms than dads that want to know that why. Was it the glass of wine I had before I realized I was pregnant? Was it, the Tylenol I took? Was it the antidepressant I didn't stop? All of these other things that they have been, really self-loathing in terms of blaming themselves for this. I can tell you so many mothers that have just had this enormous weight lifted off their shoulders when they now knew it wasn't their fault, and they can just get on to really what they should be focusing their energy on. So it's not for everyone, and I wouldn't say monolithically that everyone should have genetic testing. I think it's for some people, and it's not for other people. It's at some times in your life, not other times in your life. It's individualized, and hopefully we're doing a better job of being able to educate everyone so that they can, before they come to you in the office, before you have to have a 2-hour conversation about this, they can pre-educate and then have some really thoughtful questions for you.
Thanks, Wendy. That was lovely. Sort of a follow-up question to you, and echoing off Jeremy's comment. The question is about what degree is there hope, or is this a silly idea, that you could link the specific genetics in a kid to the treatment he or she needs, whether it's a drug or psychosocial, or what treatments won't work for this particular genetic makeup?
Yeah. So number one, we're very early days in this. So that's the first part of this. I can say already in terms of some very specific things on the medical side of things, by knowing the particular gene, I now have a much better idea of what medical issues are or are not going to be coming up, but we need to get a critical mass. And for me, my magic number is at least if we can have 100 individuals with any one of these genetic disorders, I now start feeling confident enough that I can give people advice and I can start, having recommendations for this. And we're not at 100 for many of these yet. So that's the first. The area, and I mentioned SEN2A as one example of this, I do think that's extremely, number one, I can't just look at a genetic test report all the time and even determine whether something's going to be a loss or gain of function. We actually have to do patch clamp experiments, be able to look and see how these channels are working. And I do think we're trying to get better ways of being able to do scalable outputs to understand these, to correlate that, because it is starting to make a difference in terms of treatment for the epilepsy portion of this. And I see a lot more just in terms of things that are coming through the pipeline from pharmaceutical companies. The forms of autism associated with epilepsy is where they're focusing a lot of attention, in part because they can get an EEG as a readout, and in part because I think they have things that are more proximal in terms of therapeutic agents that are more targeted in that way. So I think it's a long haul. Just to throw one thing out provocatively, which I know is not exactly this audience, but for spinal muscular atrophy, which is another neurological condition, as some of you have seen, we're now doing it here, phase 3 clinical trials in terms of gene therapy with newborns that we're identifying asymptomatically or pre-symptomatically from newborn screening and doing it on scale. This is now getting to the point that with AAV9, with other vectors that may be able to specifically work in the nervous system, long game plan, but 20, 30 years from now, one by one, I hope we'll be able to have some other alternatives.
I'm going to take this opportunity to ask one question, then I'll well, the question I had was about the genetics first approach that you showed. I was struck by the clinical phenotypic heterogeneity in that population. And I guess it made me wonder about the sort of the reverse approach that we often use of trying to identify endophenotypes with the idea of having a homogeneous population based on behavior that will lead us back to the genetic etiology of the disorder. Does that, the heterogeneity that you're seeing within a genetics first approach, does that suggest that the endophenotype approach is not going to be very fruitful?
So I still think there is value in the endophenotype, right? You still take a large amount of heterogeneity and at least you refine it to a certain extent. I guess what I've learned through this journey is that even with something you thought was relatively, so the dimensions I guess that I would say is I have seen very, very consistent homogeneous phenotypes for some conditions, but it's because I think there's a floor effect. Essentially, I have destroyed the brain, the way a brain is working so much that it's very much handicapped in terms of every single dimension. If I have a gene where there's enough flexibility, where there's a lot of potential and there are a lot of things to do, then there's a lot more diversity in terms of how that plays out. It seems like the genes very proximately will tell me about brain structure. There's a lot more direct one-to-one, if you will, endophenotype correlation there. And the closer I get to an actual behavioral output, then the more diversity I get in terms of going to that. And so I guess I would say the more biologically driven in terms of hard things that you can see, biomarkers, brain structure, other things like that, those endophenotypes I would predict are going to be more closely tied to the genetics. And once we get to behavior, there are just a lot of other things in a good way that are altering what that output looks like.
I had a question regarding the paternal age effect. So you sort of argued that that's one of the biggest epidemiologic signals that you have. And I know there are a lot of different psychiatric and non-psychiatric diseases related to, that paternal age is related to. So in terms of thinking of how it would work, the genes or the de novo mutations that are coming up in paternal age diseases, are they in similar genes that are at all related, or?
Interesting question. So just a little bit more about the biology for this, for those of you who may have been thinking about this. So the idea is that when you're talking about spermatogenesis, you're going through lots of replication, lots of cell cycles, and those obviously are just linearly increasing with age. Interestingly enough, it looks like there is actually an advantage, either a reproductive advantage, if you will, of some of those sperm with some of those mutations. They actually end up competing out some of the other sperm in terms of the percentage of cells that actually make up the sperm. So and in fact, in a very interesting way, those are some of the same mutations that drive cancer. Those same somatic mutations have a replication advantage in terms of cancers. So that's one portion of it. With that, there are then some of those mutations that are associated, as you were probably alluding to, with autism, but also schizophrenia, also with certain types of birth defects, other health conditions. And what's interesting to me is that for the same mutation, the same gene, even the same exact mutation in a different person, potentially a different genetic background, may manifest in different ways. What seems to be most consistent, though, is that the impact on the brain is universal for all of those. Whether or not they have a birth defect, congenital heart disease, a diaphragmatic hernia, other conditions that I study, that penetrance is only about 10%. And so there are probably other factors that are determining that same mutation in one host has this constellation, but universally they're all having effects on the brain. Whether or not their IQ is 100 versus 60 depends on other complicating things, but consistently, absolutely, as if the most sensitive organ in the body in terms of perturbation is the brain.
Okay. There are no other questions. I think we'll stop there. Oh, sorry. One last question.
Well, it's more perhaps just a comment on the previous question. Paternal age is related to de novo mutations and to autism and probably to schizophrenia and other neuropsychiatric disorders. But at the same time, older father's age is related to longer telomeres and reduced risk of cardiovascular disease. So it's like many things, many prenatal, or if you like, I don't know whether you call it prenatal, but exposures like this, you can't draw public health conclusions just on the basis of one particular type of outcome, which you already know, but I'm just making the comment.
Yeah,
I guess for everyone else.
absolutely. This is not the only thing, but I'll just throw out something provocative just to get you thinking. So, again, what's the news you could use from this information? And again, guys, I don't mean to embarrass you in any way. Would you do screening on sperm? Right? If you had older men, for instance, would they want to screen their sperm and see if they actually have a higher mutation load that would predispose them to some of these things? I don't know. I don't know the answer, but that's the reason why we ask the question, not to be able to play the blame game or to say that it's not other things, but we are trying to think about other things that we could use with this information for. Okay, thanks.